Okay, for the second part of the lab, we're going to measure the total amount of leaf area on the pine and on the angiosperm. So for the pine, as you can imagine, it's difficult to calculate the leaf area of a pine needle. It's three-dimensional, it has a strange shape, so we're going to use a formula that relates the length of a fascicle of a pine to the area of the needles in that fascicle. So what we're going to do is measure the length of a fascicle in millimeters. Okay, so this fascicle, 52 millimeters in length. So what we do then is take that 52 millimeter length and plug it into the formula in your lab handout, which states that the area in square centimeters of this fascicle is its length, which is 52 millimeters, multiplied by 0.1005. After we get that number, that tells us the area of one fascicle. The next part is fun. We have to estimate how many fascicles there are on the tree. Frankly, these trees are small enough that you can just brute force it. You can count every needle, every fascicle on the tree. So I'm going to get started on that. Now for broadleaf trees, it's a little bit easier to calculate leaf area because their shape's a lot simpler. So for this lab, we're going to calculate the total leaf area by measuring the area of 5 average or typical leaves. And we're going to do that using this dot grid. The dot grid just has a series of squares on it, each of which is 1 square centimeter. And each of those squares has a dot in it. So we then take that grid and put it on a leaf and count the number of dots that land on the leaf. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 dots. So that leaf is 15 square centimeters. What we do is then repeat that for 4 other leaves on the tree and calculate their average. That's the average individual leaf area in square centimeters. And then count the leaves and multiply those two numbers together, and that's the total square centimeters of leaf area on the tree.